Hello, welcome to the Colors for Crafts podcast, the podcast that aims to educate, inform, and engage our audiences about the meanings of color, textile and craft coloration practices, and the future of color through research, education, and experiences stemming from the Colors for Craft project based at the University of Helsinki. I am your host, Dr. Dustin Neighbors. In this episode, I am joined by Dr. Juuso Laitila to talk about the chemistry of dyeing. Join us as we discuss polyphenols and tannins in analytical chemistry of dyeing and the role of this research in the Colors for Craft project. Please allow me to introduce our guest. Dr. Juuso Laitila is a specialist in the analysis of polyphenols and other natural products. His research with natural products spans more than a decade, extending from his early studies as a bachelor's student all the way to his current position as a postdoctoral researcher in the Department of Chemistry at the University of Helsinki. Dr. Laitila is fascinated by the chemistry of nature and how many intriguing natural compounds are abundantly available and present in plants, even in urban environments. In his current role, he is studying the usage of natural compounds as natural dyes and textiles. In Colors for Craft, he is motivated by the multidisciplinary nature of the project, where chemistry meets history and craft sciences. Juuso, welcome. Let's start with the first question. What is your research background, and how did you come to study chemistry and natural dyes?
The story of me getting involved with chemistry, it is a combination of deliberate choices that I have made myself, but there are also a whole bunch of coincidences that have happened over the years. So I finished my high school back in 2012 in Rovaniemi. Already in high school I was really interested in natural sciences, so in mathematics, in physics, in chemistry. And at the time the guidance that you got at school was that if you're interested in these subjects, you might want to consider pursuing a career as a medical doctor. You might want to consider applying to a medical school. Yeah, I was one of those people, and I did apply to a medical school, and to medical school in Turku, to be more precise. I didn't get into the medical school on my first attempt, but I did get to the university to study chemistry, which was my second option. And I was interested in natural sciences, yes, but the idea behind studying chemistry in the university really was that, this would give me an advantage when I applied to the medical school a second time. And I did apply a second time, but I really didn't put too much effort into that attempt, so it barely counts as an attempt. And this was after my first year of studies at the university. Maybe the idea wasn't that clear after the first year, but definitely after the second year it was very clear to me that I will pursue a career with chemistry. It turned out that chemistry is actually really, really interesting. Within chemistry there's obviously loads of different lines that you can, or different subjects that you can study. But especially the chemistry of natural products was the one that really intrigued me. And I was really lucky to be able to land a summer job at the Department of Chemistry after my second year of studies. I got a summer job in Natural Chemistry Research Group, and as the name suggests, it's a group that focuses on the chemistry of nature and also in all the related analytics. It started as a summer job, but I then ended up doing my bachelor's degree there, my master's degree there, and eventually I did my PhD thesis there as well. I do remember it very clearly. If I go all the way back to the second year student or second year of studies, I do remember it very clearly that the idea that really intrigued me and really motivated me was that if I pursue my studies in this research group that focuses on natural products, I will gain better understanding what's actually happening in the nature all around you. And even in these urban environments, there are whole loads of plants there that are producing really interesting polyphenols and tannins and other natural products. And by studying that kind of chemistry, I will also gain the tools that then enable me to do my own research and see how things are for myself. That idea is still with me today, I would say. Still a very big motivator for me. my studies have and my research has always focused on the chemistry of nature, and especially in these compounds called polyphenols and tannins. And I'm really glad that I got this job as a postdoctoral researcher in this Colorful Crafts project, because it really lets me return to my roots as a chemist.
So that was the original interest, was plants.
Yeah, definitely. Okay. Yeah. Natural Chemistry Research Group, they study the chemistry of plants.
Yeah.
And polyphenols and tannins mainly. I think we will touch upon those later on.
Yeah.
But yeah, it was about the chemistry of plants, chemistry of plant kingdom. And that's a fairly unique group in Finland. I don't think there's another one like that at the moment. Mm-hmm. Like there are individuals who do research on natural products and on the plant chemistry. But like that type of thing, and like that you have an entire group focused on that, I don't think you have another in Finland. So that's one of the coincidences that I mentioned. That was like the original interest, like why I pursued the studies there, and I'm still on that track. I have had a few I have worked outside of academia, which a little bit as well, but within this project, I can really return, the roots of my interest in chemistry. That's kind of neat.
So that's one of the interesting things, because you're talking about your interest in plants and, this kind of, I guess, passion to pursue this. All I see is little Juuso looking or on the ground, watching the plants and looking at the plants. Yeah. That's the idea that I get, because I think that's something when we think about people working in nature or working with nature and the environment and looking at plants, that's what interests them. What is it, if I can ask for a little more specificity, what is it about the plants? Is it how they grow? Is it what affects their growth? What their environment is?
It's really about the chemistry of the plant. So, this group that I'm referencing, and they study especially the secondary metabolites. So for instance, these polyphenols and tannins that we will talk about later. So it's not about directly measuring the growth, for instance, of a plant or something like that, but it's about these chemicals are related to the chemical defenses of the plant, for instance.
Yes.
So these compounds are important, in the chemical defenses, like I mentioned, but also attracting bees and others, protecting the plants from abiotic stresses, like radiation, for instance. So these are the type of compounds that that group studies, and I don't know why I got this fixation on the tannins and on the flavonoids and on the polyphenols. I don't know what really triggered that, but that's the type of chemistry that I'm really interested in the plants. And those are the type of things that I am now studying as well, but it's from a completely different perspective.
I think that was really interesting is when we work on certain things, and I never thought that I would work on hunting, historical hunting, because I didn't like it as a kid.
Yeah.
And it's funny, as we get older, what we're drawn to. You talk about tannins being the thing that kind of piqued your interest. And I was reading about your research as well. You began looking at red wine.
Yeah, that came in at the master stage.
Okay.
The in as part of the studies, you do these laboratory practical things. As part of your master studies, we had this 5-month period of lab work. And before that as well, we had 2 shorter periods. Yeah, in my bachelor studies, I did this laboratory practice period about the tannins and flavonoids in meadowsweet. I'm not forgetting the
meadowsweet. Yeah.
Not meadowsweet. I actually forgetting the English translation of the plant. Fireweed. Yeah, sorry, fireweed.
Okay, fireweed.
Yeah, fireweed. Yeah. So I isolated the main tannins from the leaves of fireweed and also the main flavonoids. And my supervisor of this bachelor's lab practice was doing his PhD about those same things. But yeah, that's what started it. So started by isolating the tannins and flavonoids from fireweed, and I have now actually returned to fireweed as well. We have studied that this summer actually.
Oh, nice.
There's some kind of circle there as well. But then I did the more advanced lab practices I did about the alkaloids, which are certain types of natural products. But yeah, those I studied in tropical ficus species.
Okay.
And then I landed on the red wine thing.
Because I was wondering if it was one of those moments where you spilled red wine and it stained, and then you're thinking, oh, how did the chemistry... Like, you think about how people, when you read things, like, how did you get onto that? But I like the idea that you talk about there's different elements in nature that you look at, and you then process of interrogating the actual ingredients itself, the compounds themselves, and how they react to being put with other chemicals, other things like that.
I gotta still mention about this because if you list like the things that I've studied, like the fireweed, the ficus species, red wines, now this plant dyeing stuff, for me there couldn't be a more clear connection between all of these, because when I talk about these compounds, I have these molecules popping in my head that this plant has this compound and this has this compound, and they are all repeating. Sources.
For someone outside of chemistry, I don't see that connection. They're like so different. They're like wine, plant, okay. But then you're sitting here, and this is the interesting thing, because you're saying there's a common compound in each of these.
Sure.
Interesting.
Yeah.
Okay.
For me, there couldn't be a more clear or bigger connection between all of these. Okay, maybe the ficus species are a bit different, because those like fireweed, red wine, and then everything that I'm now doing, it's about polyphenols. Alkaloids and polyphenols are like different groups of compounds.
Okay.
So maybe the ficus species are a bit, separate thing in a way, but it's still all very much connected in my head.
Well, that's a very good segue, because the next question I wanted to ask you, what does your current research focus on? So what are you working on?
Yeah, so now I'm working, like if I want to put it shortly or briefly, I would say that I'm working on the chemistry of dyeing. Color for Crafts in general focuses on the chemistry, natural pigments and natural dyeing. I'm very much involved in the chemistry part of that. And it's like what I do is also very much like basic research about the subject. So I would like to describe this like as a structure-color connection study type of thing, because chemistry dictates what kind of colors you get from a certain dye plant. That's maybe the biggest thing that I'm now currently focused on, studying the chemistry of different dye plants and seeing what kind of colors that gives you, and trying to, bridge this chemical composition into the colors that you get. So it's like I'm trying to understand like more deeply what compounds you have in the dye plants, like their structure, how that structure then is transferred to the color. So it's like structure-color relationship.
So it's not focusing on how the actual dye adheres to fabric. It's more about the process that you have to make it into a dye from the plant itself.
It is that as well, because if you imagine that you have a dye plant and it has 200 components in it.
Okay.
And then you throw in a piece of wool. Like not all the compounds interact the same way with the wool. Some are, some remain in your sample. They are not absorbed to the wool at all, while some are. So doing the actual hands-on dyeing with the dye plants is a very important part of this, because if you see the chemical composition of a plant, even if you can characterize all the components in there, you don't know which are important for the color unless you actually do the dyeing experiments as well. Then you can see like what is absorbed to the wool and what is not. Then you can also try to back extract the components from the wool to see what you find in there. Whole process that I'm interested in. It starts with characterizing the compounds you have in dye plants, but it ends up in trying to understand what's then actually in the wools that you have dyed.
You said a second ago that there's over 200 compounds in a specific type of plant. Is that the same for all plants, or is there some plants that have more compounds?
Yeah, that was just a rough example. Rough example, really. Yeah. But when you are talking about these polyphenols, that most of the natural dyes are polyphenols. So if you consider those, the number of compounds that you can have in a plant, it really varies from, let's say, from dozens to hundreds in a single species. So there's a lot of variation there.
Does that then mean if the more compounds there are, the more research is involved to identify each of the compounds?
The more difficult it becomes.
Okay.
If you only have 10 compounds,
you're done.
Yeah, I mean, that's a piece of cake, walk in the park.
Yeah,
but
okay.
if you have 700 of them, it's a completely different thing. But it's very hard to give a number on how many compounds you have in a plant, because you have like quite limited number of main compounds that make up most of the mass of the plant or the plant extract. But then you have a lot of these minor compounds there as well. It's really hard to give a number on how many of those you can have, and it really also depends on the chemical composition. Like if you have certain types of tannins in a plant species, then you know for a fact that you are working, you're talking about hundreds of compounds easily. So it depends on the chemistry as well.
So why did you choose the hardest one with hundreds of compounds?
I mean the tannins, they are like historically a thing in the field of like natural compound chemistry. But if you don't if a researcher doesn't understand something, they don't understand why something is happening, it's always the tannins. Just blame, just blame the tannins. And they are compounds that traditionally, like, people have tried to get rid of when they study like plant chemistry.
Well, this would be a good point when we talk a little bit more about the tannin. But one of the things that struck me as you're talking about the plant and then looking at how maybe it adheres, but also the different compounds and all of this stuff, it makes me ask the question of what is the methodology and process. So is this what we would consider analytical chemistry, where you're taking the process and what does that process involve?
Yeah, I mean, this is very much about analytical chemistry. So, if I would try to get a job outside of academia, I wouldn't advertise myself as being an expert in the chemistry of plants. No one cares about that outside of academia. No one cares about the tannins. I very much advertise myself as an expert in analytical chemistry. So that's the tool that lets me study things for the chemistry of the dye plants. Yes, a big part of that is the instruments that you use. You have to be able to understand those, but then also your whole process is an important part of that. As an example, like back in Turku, back in the previous research group, like I was taught to handle the plant material so delicately, take so many precautions that you do not change the chemical composition when you extract the compounds from the plants. Everything remains exactly as it was in the plant itself. That's the way I was taught taught to handle the plant material, and then now I'm boiling the plants to get this dye sources, or to get these coloring solutions. That's where the analytical chemistry starts. So it's really the whole process, because it doesn't matter how good or how good or great your analytical instruments are. Everything starts from extracting the compounds from the plants. So in that way, it's like analytical chemistry is a process. You have to understand like what you're doing to your source material, so that you can then interpret your results. Because everything's connected there. So it starts with extracting the compounds from the plants by some means. Now I'm boiling them because that's the application that I'm studying. It's the traditional way of getting the traditional way of doing this plant dyeing thing. You boil your plants. So that's now my extraction method. But then you sort of have to think the path or the journey of the compounds, from there until you have analyzed them with your fancy instruments. So, that's a process in a way. But the analytical side of things is very heavily then also focused on the instruments that you are using. So I'm doing what's called liquid chromatography, mass spectrometry, spectroscopy. Those are the more precisely the tools then in the end.
You raised 2 points that now make me want to ask 2 further questions. The first thing is you talk about the last research group you had and being protecting of the plant and all of this stuff being very fragile. As an analytical chemist, if that's the right word, do you go out in nature and actually get the plant, or do you have to get it in a specific form before you can study it? Like, does it have to be a powder form? Does it have to be, how do you receive that particular, and then what led you to the point of boiling the plants? Like that's an interesting point there.
Yeah. So that's one of the perks definitely with this line of job. When you study the chemistry of plants, where do the plants grow? Well, they grow in the nature, so you go after them. Now we've been collecting them fresh, and then we have also processed them fresh. So there's no drying, there's no powdering them or anything like that. We just take them, we weigh them, we boil them. But that's, if we again reference back to the group in Turku, they're getting a lot of advertisement here. Rightly so. But, there it was the research questions were completely different. So there the plants were collected, they were immediately frozen, they were freeze-dried, which means that, they are dried under high vacuum and in cold temperatures. That's the best way to preserve the chemistry of the plant. Once you pick the plant, if you pick a leaf from a tree, there are all these enzymes, for instance, in the plant that can start, degrading the chemical composition. So the best way, if you really want to preserve the chemistry, is take the plant, maybe dip it directly to liquid nitrogen, so that it's flash frozen.
So it freezes them
immediately, and then you freeze dry, and then you have your well-preserved sample where the chemistry is intact. And that methodology was necessary for the stuff that they did in Turku, but for me, my application is much more hands-on, because we are studying the historical recipes, and the whole context for the analytical chemistry comes from the historical way of dyeing wool with plants. Boiling the plants was the main thing.
So have you had any experience with looking at historical recipes or working with historical documents and references and practices?
Yeah, I have I have a colleagues in this project who are historians who really look much more deeper into the historical recipes. And when I say that the boiling used to be the thing, they will probably get shivers when I say this, as if there was only one recipe that was followed. So there were loads of recipes that existed with all these minor twists and details and all this. If you now imagine that we screen 70 plants and we want to compare the chemistry and the result that we get from the 70 plants, we cannot have 5 recipes for each of them.
Yes.
So you can start calculating how that would add up. Boiling the plants has been generally the main way of preparing the coloring solutions from the dye plants, and we have to do that to keep everything comparable in this project. But definitely it would be also at some point really interesting to study the different recipes with individual plants.
And looking at how they are affected in terms of, okay, if this recipe says this, this, and this is the reaction that happens here. Where if this recipe says, add a little bit more of this, but you get a completely different one. And I love the idea of that, but also my brain sits here and tells me that's a lot of work to recreate all those recipes step by step, and it all produce different colors or different shades or...
Yeah, it's maybe more about, if you have a dye plant and then you have these different recipes, this is my hunch, that you are mainly talking about different shades in opposition to, completely different colors. There are, if you have a dye plant and you boil it, you get a certain color, but then there are recipes where you ferment the plant or the extract. That then gives you actually very different color. But if you have these variations of the boiling method, I think they will more or less give you the same color in a way, different shades for sure, but especially from the chemical point of view, the compounds are still mainly the same. Just different proportions, maybe different degree of degradation of the compounds, and when you degrade the compounds, the properties of the compounds change, and that affects then the shades. And
that was something that at our last meeting that we were all sitting there, I hadn't thought of that. I hadn't thought about if the plants are peaked at a specific time of the year, close to winter, then a lot of those compounds have already been frozen because it's getting colder, the weather's getting colder. That can affect the certain shade. But also, if the plant is damaged, and you don't know if it's damaged necessarily just by looking at it, but as you said, the compounds are already in degradation, that will then change how the shade is as well.
Yeah, and seasonal variation is very much a thing with chemistry, like with polyphenols, with tannins. The compounds might be more or less the same throughout the growing season. But with seasonal variation, you get like variations in the concentrations and proportions of the compounds in the plants. Start off with certain composition, like 5 times more of compound A compared to compound B at the very early summer, but then things might be vice versa, at later stages of the growing season, September or.
Now we've talked about that you pick the plant, or you grab the plant, you either liquid nitrogen, you freeze it, or it goes boiling.
Yeah, we don't do the freezing.
We don't do the freezing.
It's
an example.
Yeah, exactly.
So you have that. What's the next step then? Do you then take it and look under a microscope? Is there a certain machine that you use?
Yeah. So we don't really do like microscopy, and I don't want to go into too much details about individual experiments, but I can expand on what I said earlier about the chromatography and mass spec and all this, because that's where everything ends up eventually. Yeah, if we have a boiled extract or we have a sample from any other source, the chromatography, mass spectrometry, and UV spectroscopy are the analytical tools that can tell me what compounds we have and how much we have them in my sample. So, like I said, I previously referenced that you can have hundreds of compounds in a plant, in an extract. So the first step of the analysis is the chromatography side of things. So chromatography in general is a technique that allows you to separate your individual components from the mixture. So it's an instrument, and if I inject my sample there, let's say 5 microliters, all the compounds are stacked in one small volume. But then throughout this analysis, they are separated by certain mechanisms, so that they come out of the instruments one by one. Okay. Essentially.
And that allows you to look at them individually.
So when you don't have 300 compounds overlapped, but they come out of the system one by one, it's much more easier to identify what they are. So chromatography is like the separation technique. Very many different variations of that, but generally chromatography is used to separate compounds.
That makes sense to me as someone who is not a scientist, not science-minded at all.
Yeah.
How long did that take?
Depends on the instruments that you have, and also your sample. Like if you a sample that has only 3 components, it's easy to develop method that's very quick because you don't need to separate very many compounds. With the plant extracts, it's can vary depending on your instrument. Let's say from a few minutes to 20 or so minutes nowadays. Okay. Like back in the 90s, when these instruments were first developed, from 2 to 4 hours, for example. Okay. Now we're talking about minutes. So there's there has been a lot of development in these chromatographic instruments, and it's very good that we are talking about minutes nowadays, because if we have hundreds of samples, then it's possible to actually process that many samples.
So because that was the thing that I learned about this summer, because I was Leaf, who was on our project with us and worked with Krista, and they're doing the experimental recipes. They talk about it can be several days up to a week before they can actually get the dye to a certain point that it can adhere to the fabric, all of this. So in my mind, I'm thinking, well, if that's the case and you've got 200, it's going to take you days to process. But interestingly enough, it can take only a couple of hours.
Yeah, I mean, I'm only interested in the stuff that the plant has prepared for me. So my life is much more easier compared to people doing synthesis. Yeah. And also, like, people, I think they're also, they might be doing some modifications or something like that. Like, I'm taking what the plant is giving me, so I don't need to do any modifications to the sample. So I'm just interested in what's in there already. So for me, like, sample preparation is, so if you would now start a timer, there's a tree growing outside the window. I could go there, grab the leaf. I could just very quickly dip it to some solvent and take a sample and inject it, and I would be, I could tell you something about that plant in, let's say, half an hour or 45 minutes. Okay. Yeah, but that's, again, you come to if you think about this analytical process, then you need to understand that I'm only dipping the sample to a solvent, so I'm only looking at the compounds that are, on top of the leaf. So I'm not getting the full picture of the plant. I'm getting what I'm getting by just dipping it to a solvent. I'm not, seeing what's actually in the plant cells. I'm just looking at the compounds that are on top of the, like some waxes or something like that, maybe.
Okay.
But I could tell you something.
You could tell us something in 30 minutes. Yeah. It sounds like this is a process that reveals a lot of information and that could be developed or give us information that can then be developed. What, in your opinion, makes analytical chemistry important? Why is it relevant today?
Well, it's whenever you ask, like, what's in that, or how much of, something is in, whether it's a plant or some, soil sample from, or water sample from the nature, like, whenever you ask, like, what's in there and how much of that do you have, like, it's always the analytical chemistry that then can give you the answer.
I will never ask those questions because that never crosses my mind. Every day I'm not like, what's in that? What made that? I'm more like, where did that come from? Who had that last?
The downsides of, like, depending on your background more precisely, but the downside of being a chemist is that you see maybe a bit more in depth into, at least you think a bit more in depth, like what's in everything.
Yes.
So
I do have a quick aside question, because one of the things that I love watching is like political thrillers and all of this stuff on TV. Do you ever watch a show and someone's in a lab analyzing something? Do you ever go, that's not right? That's wrong?
Constantly. Constantly.
Because I'm the same way with history. Like when I see people like doing something like, that's not the way you do it.
Constantly. And there's like one case specifically that comes to my mind. It was this Finnish TV show called Ivalo.
Okay.
And they had some laboratory there, and there was a person working in there. And I was like, "You have to be kidding me! That the budget for this was kind of big. So why couldn't you hire like someone who has sometimes seen a lab, or done actually something in the lab? Like some of these sets that you have in the TV shows and the practices that they do.
At least do something that's a little more close to reality.
But I mean, I understand fully that it's if it works for 95% of the people, so why would you care about the one chemist that's going nuts watching the show? But and I guess that happens every time when you have in TV shows you have these hackers or IT geniuses who are just slashing on their keyboard and I'm 100% certain that if you have an actual IT expert watching, they are losing their minds watching that. But about the analytical side of things, actually, I think we can still expand on that a little bit, because I was talking about the chromatography and how you separate the compounds with chromatography and they come out of the system one by one. But that's still there's still two techniques that are very important for me. Like analytical chemistry is a very wide subject, but if we now limit to what I'm doing, then it's we can actually talk about this in a time span of a few hours. But that doesn't still tell you about the compounds that are popping out of the instrument. So that's where the spectroscopy and mass spectrometry. Basically, mass spec can be described as a very, very precise scale. So it's an instrument that can give you the mass of the compounds that are coming out of the liquid chromatography system. The instrument that I'm using, for instance, it's called a high resolution mass spec, meaning that it can give you the mass very, very, very precisely enough that you can calculate how many carbons, how many hydrogens, how many oxygens you have there, so you can deduce the elemental composition. And that's a very important tool in characterizing what compounds you are seeing. Then the UV and visible light spectroscopy is another important tool. Again, when this compound is coming out of the liquid chromatography, then it goes through this spectroscopy instrument in a way. With that system, you can tell how the compounds are absorbing UV light and visible light. So they can, for instance, tell you that how the compounds would look to a human eye. If I see a compound coming out that absorbs light at 430 nanometers, for instance, it then tells me that the compound is yellow, or maybe a tad of orange, with a tad of orange maybe there in the mix. But it gives you information about how the compounds absorb light. That's of course very important for when you study dyes.
Okay.
But in general, polyphenols, like different groups of polyphenols absorb light in different ways. And then when you know something about those patterns, then you can immediately like which group of compounds this compound now belongs to. And when you combine those two, you get like the mass and you get the way the compound absorbs light, then you can already say a lot about the structure, like more specifically what type of compound you have. And
so you reached that point. So you've got the plant, you've boiled it down, you've got the extract, you've put it into the spectrometer. Now you've got the information. You know that you've got, a yellow here. What do you do with that information then? Where does it go next? What does it lead to?
Well, that is the information that you can then use to try to understand why the plant gives the kind of color that it does.
Okay.
So, and at this point, like I said, this is still very much about basic research. The chemistry of dyeing is not, I don't think it's like very well understood necessarily. When I talk with people who do a lot of this stuff, like who practically do a lot of this stuff, I hear all these like generalizations that a certain compound gives you a yellow color. They might say that a quercetin gives you a yellow color, and a quercetin is a type of a polyphenol. It's a flavonoid. Yeah, quercetin itself might have like a yellowish hint to it, but then in nature you rarely find quercetin as such in the plants. You have the quercetin, but then you have all these different carbohydrate units attached to it.
Okay.
Typically, glucose is a very typical compound. So you have this core structure, you have your quercetin, but then you might have one glucose or two or more glucoses. They might be attached to the same positions or different positions. And then you have different carbohydrate units, galactose as well, rhamnose, for instance. And when you take all these into consideration, then you instead of having quercetin in plant, you can have dozens of different types of compounds with quercetin in the middle. And all of these are different compounds. They have different structure, even though they have the same kind of building units, but the structure is different. When it comes to, the chemistry of dyeing, chemistry in general, structure is everything.
Yes.
So, for when someone says that the quercetin is a yellow compound, I'm wondering like, okay, what about these 27 quercetin derivatives? Like, are they all yellow? Are they all yellow, or do they have differences when it comes to dyeing with them?
So essentially, in the end, you're looking at trying to identify. You're taking the plant. You're trying to identify the structure, and basically the answer at the end, especially in relation to dyeing, is this is the structure. This is what you're most likely going to get.
Yeah, I mean that would be
the idea.
That would be a great outcome, to be able to link the structure more precisely to the color that you get. Okay. I very well understand where these generalizations come from. It's a difficult topic.
Because a lot of times they're based on assumptions
rather
than actual facts.
Yeah. There's a lot of that as well, and the analytical methods might have been such previously where you're sort of breaking down all these quercetin derivatives into just quercetin, and then you measure that this sample has quercetin, and then you make the conclusion that quercetin gives you yellow, which is not wrong. It's just then you have limitations in your analytical tools, and you don't see the entire big picture in a way. So I fully understand where that comes from, but for me it just raises so many questions about the more precise structure. Again, there's an images popping in my head about these molecules, okay, how does this structure compare to this, even though they're both quercetin derivatives? And I don't think that kind of information exists too well yet, and especially if you go to the tannins, that's a whole different thing altogether. There it's even more vague what people mean when they say that this is tannin containing compound, and that's why it gives you some colors. To me, it's they're saying water is wet. There's very little details there.
Yes. One of the things that just popped into my head that I wanted to ask you, do you see things in molecules and compounds? When you look at color, do you think, okay, I know what that color structure looks like, or that compound looks like?
No, not definitely. No, not if I'm looking at a certain color. There are too many options there. But whenever I'm talking about these compounds, I usually have things like example structures in my mind. Like, if I'm talking to you about condensed tannins, I cannot avoid of thinking about condensed tannins. Then I do have like structures in my head when I'm talking about this stuff.
We had the Arcticum exhibition, and it was the model of the molecular structure. So that the reason why it entered my head is that what you see all the time is that molecular structure.
In a way, maybe so. Like, that's actually a good way to say it. Maybe it was like a sneak peek to my head or something. Yeah. That was, by the way, a very fun thing to do. This mobile of the 3D structures. It was a lot of fun. But
I thought that was a fantastic idea. Like, I would have never come up with that, but that was really cool the way you did it. And what did you use? Was it the type of material? Was it like a toy or?
The models were 3D printed first.
3D printed, okay.
It was just plastic. I don't remember the plastic. I think it was PLA or something like that. I don't remember too well anymore. I did that at the workshop here in the Kumpula campus. So the kind people there, printed the things for me based on the models that I made. But then from there on, it was just, hands-on painting the models, acrylic paint and brushes.
And if
also
our listeners,
some sanding was involved.
If our listeners want to know the molecular structure that we're talking about, you can go on to the Colors for Craft website and look at the digital exhibition from the Arcticum, and there are photographs of the molecular structure, which, again, it stands out to me as one of the great pieces from the exhibition.
Yeah, and I mean, my role in that was the models themselves, like the space where the mobile was, like where the models were, like the space, and then the decorations, they were done by other people.
Yeah.
But all together, I would say that the room was quite amazing.
Yes.
With the black walls and the colorful models and the spotlights, and then the wall rugs and the bright plants and everything. I think that was.
It was kind of like almost an immersive experience because of the black wall, because you had the mobile, it was hanging, and then you had the textures on the wall and the colors on the wall. It was it was fantastic the way that they did it. But I do still think that if you had your molecular structure in the room just hanging, even that is something to see, like, this is how we see colors in some ways, or the molecular structure of that.
Yeah.
Talking about compounds, talking about what you were doing, you keep saying polyphenol. What is polyphenol, and why is this an important chemical substance, if that's the correct word?
Yeah, that's most definitely a correct word. Yeah, so polyphenols, like, plants make primary metabolites and secondary metabolites. So the primary metabolites are the carbohydrates, they're the proteins, they're the fats, everything that the plant needs to basically grow and live. And they would manage, like, in a completely isolated system from, where no outside threats would, do any harm to the plant. They would survive with just those, maybe. But then the secondary metabolites are the compounds that are then needed in the actual living world, in the actual nature. So they are the protective compounds that protect the plants from the herbivores, from the radiation, from the drought even. They attract pollinators and all these sorts of things. Yeah, so you have your primary and secondary metabolites. And actually, my professor in Turku, like, really wanted to skip this term secondary metabolite and wanted to, really heavily lobbied for this term specialized metabolites.
Oh, okay.
And I do see his logic very clearly, because saying that they are secondary somehow implies that they are, less relevant, whereas in fact, like, they are very much needed for the plant to survive. When you have limited resources, the plants don't make these compounds for, just for fun, and they can be a significant proportion of the mass of the plant can be the secondary metabolites, and at least the extracts that you get from the plants, significant proportions of those can be secondary metabolites. So now we have the primary and secondary metabolites, and in the secondary metabolites, again, you have different main groups under that. So you have your terpenes, you have your alkaloids, and then you have your polyphenols.
Okay.
So polyphenols are one of the main groups of the secondary metabolites. They are perhaps the most abundant ones in the nature. Alkaloids are, they are much more rarer. And I would say that the terpenes are then somewhere in the middle. Polyphenols are the most abundant ones. And they are compounds that are basically built of hydrogen and oxygen.
And is that why that's what they're focused on? Not to say it's easy, but because there's more accessibility to it, because there's more, or
I
is this one of the main kind of...
it is down to that as well. And like in this, if we're specifically talking in the context of plant dyeing, most of the dyes are polyphenols.
Okay.
So that's why we are focusing on the polyphenols. There are compounds that belong to the group of terpenes, like carotenoids, like the orange pigments in carrots, for instance. They are terpenes, but most of the compounds, or like most of the dyes, belong to polyphenols. So that's why polyphenols. And again, you can go further within the polyphenols. You have again different groups of compounds under there, and we are talking like number-wise. We are talking about thousands of compounds.
Yeah.
We're talking about just polyphenols.
And just to clarify, so when we say dyeing, and we're saying we've got a dye, for it to be a dye from a plant, and I'm just asking to make sure then it has to have polyphenol.
No,
and if we don't get a color, then because it lacks polyphenol?
No, that's a good thing to ask. So not most of the polyphenols actually are not pigments. They are not colored compounds. Okay. So for a human eye, they are not colorful. Most of the polyphenols are like that. There's actually a very limited number of polyphenols that are pigments, or dyes direct. But in these plant dyeing processes, even compounds that are colorless, you can get a color out of those as a result of the plant dyeing process. Okay. And we can touch upon this later as well, but this comes very much down to the way you prepare your fabric that you're dyeing. Do you like pre-process it somehow with mordants? So these mordanting compounds are very important to bring out the color of the polyphenols. But yeah, most of the polyphenols actually, like tannins as such, they are not pigments. Most of the flavonoids, they are not pigments. Okay. And that was very hard for me when I started actually in this project, because people were talking about compounds that I have isolated myself, and they have like white powders as pure compounds. And they're talking about them as pigments. And my head is just exploding. Like, that's not a pigment, really. But I have learned that these pre-processes that the fiber, like the textiles, the pre-processes of the textiles are very important. Get the color in the end. We can talk about that later still.
I think that's one of the exciting things about this project is the fact that we're learning cross-discipline terminology. Yeah. And having that communication, because what you would call a pigment is not what someone who is an experimental historian or researcher would call a pigment. And I think that teaches us to clarify, to be specific, but also to understand that there's variation when we say certain things, and it's always good to clarify.
Yeah, and I mean, definitely we have had, like, it has taken, a long time to be able to understand one another on the same level, because of course my background shows when I'm thinking about plant dyeing. Like, I think it from my previous background, like naturally. And when people are doing like this practical dyeing experiments, replicating historical recipes and all that, they have their own background and they are thinking about things from that respect. And then you just have to try to find the middle ground there, because I wouldn't have anything to research if it wasn't for the historians. Because they are the experts in what all this historical stuff. Like,
can I get you to say that one more time? I wouldn't have anything to research without the historian.
Yeah, I can. Yeah, but that is very much true, because I wouldn't know the context of my research without the historians, because this is, I say to people that the research that I'm doing has cultural historical implications. I wouldn't be able to say that if I didn't know that from the historians.
I think that's one of for me, that's the exciting part of this, is because for so long it's always been sciences, humanities. Yeah. The two shall never cross. But there's so much to learn between the two disciplines. And I think there's, and forgive me, there's so much emphasis in funding for the sciences, not realizing that actually the humanities play an important role in contextualizing, in giving more information, background to this, And it's great to see, and I think this is one of the great things about the project is that we are seeing where the two come together, and it's really fueling this understanding and this innovation and sustainability that we didn't have before.
Yeah, and for me as well, like, the colleagues in this project are showing me ways to utilize plant chemistry I didn't know that was really possible. Like the exhibition that you talked about previously, Colorful Crafts.
Yeah.
Like there were loads of just amazing pieces out there on display, like that were dyed using plants, like all these wall rugs, and like they were just amazing. Like I would never think that they were, dyed using plants.
But that was what was so cool about it, that huge space. And you look around and looked at everything was dyed with plants.
Yeah, I mean, that,
naturally.
Yeah, that was just amazing. Yeah.
Yes. And it's one of the things that if you do get to see the digital exhibition. So you brought up tannins. So now we talk about tannin.
Yeah.
Why tannins?
Yeah.
What is tannin?
Yeah, I can continue where I left off with the polyphenols.
Yeah.
So, polyphenols again are distributed to several different main groups. Flavonoids is one of those groups, and tannins is another. Yeah, tannins, like I said, historically they have been a very challenging group of compounds to study because the analytical methods maybe have not been.
And just to clarify, tannin is not a plant. It's a compound.
Yeah. So tannins are specialized metabolites belonging to the group of polyphenols. So they are chemical compounds that the plant makes. And they have had their fair share of historical use cases, like, I don't know if this is the correct word in English, but like tanning leather, like you use
tannin leather.
Yeah, that's maybe the most famous example of historical use cases of tannins. Yeah, but again, everything can be always dissected to smaller parts. So within the group of tannins, you have different main groups yet again.
Okay.
You have chlorotannins, which we can discard quite quickly because you mainly find those in algae. So they are not really present in terrestrial plants. And then you have hydrolyzable tannins and condensed tannins, and they are very different groups of compounds. Okay. Both are maybe, we can talk about molecular weight, which is the weight of individual compounds. Both are relatively high in molecular weight, which separates them from many other polyphenols. But I think that's pretty much where the similarities end. Like structure-wise, they are very different. The biosynthesis of them is quite different, and the properties can be quite different. But it's with tannins, it's usually people claim that tannins have certain types of properties, and that is very, very big generalization because we are talking about hundreds and even thousands of compounds. So saying that tannins have this property, okay, some of them do, but some of them don't. Again, it comes down to the structure.
And that's why you said earlier that tannins are a problem because there's no consistency.
Yeah, like when people say that this color is now produced by the tannins, I don't really understand what they mean because there are like hundreds and more options in my head. Yeah. Which structure-wise are very different from one. That's the basic issue, when you talk about tannins. But that's also one of the things that I really want to try to get a clarification on. Because I can bring in, the chemical understanding about the tannins. And I don't want to, just discard what people are saying when they're talking about tannins. No, I just want to take that as a research question and try to see, what do they actually do? And what's the actual tannin composition in, let's say, some birch bark, for instance. It's a quite well-studied plant material. But anyway, I have the ability to really answer the question, what type of tannins we have in a dye plant, what happens to them when you put them through to the dyeing process. One of the key things that interests me in this project. But yeah, we're talking about very broad group of compounds when we are talking about tannins.
And in this particular case, because and it kind of maybe leads to this next question, which is when we're thinking about tannins and you're studying them, there is the complication that there may be inconsistencies or it differs in many ways. What is the goal then, from the tannin to identify how to make them or utilize them in a dyeing process to make them more sustainable, to create processes or methods of extracting it more easily so that it can be utilized? The final question I have is, how does your research, analytical chemistry, polyphenols, and tannin contribute to the Colors for Craft project?
Yeah, so it is still very much about, the basic research of trying to understand like what is the role of tannins actually in the color plants. And with tannins specifically, I know that there would be other people in the project who would be much more better to answer this. But anyway, like with tannins, I know that tree barks, which many of them are tannin rich, more specifically rich in condensed tannins. Like for those, you have different types of recipes. You have the boiling, but then you do also have these fermenting methods. They very much change the tannin composition, and they change the color as well. That's one of the questions that would be nice to be able to answer, like what happens to the tannins when they go through different dyeing processes. And you can, with like fermented tannin containing plants, you can get colors that are quite difficult to get otherwise maybe, like reds, for instance. I think red is quite difficult color to get to in a dyeing process. But yeah, like in general, my contribution is, like in a bigger picture, I would say that my contribution is to bring like exact language to when we are talking about tannins and different polyphenols. Because of my background and my expertise, like I can ask questions that other researchers might not even realize to ask rather. Tannins do this, but I can press that issue further, like what kind of tannins are we actually talking about? What happens to them during the dyeing process?
So to clarify and to better understand tannins.
Yeah, because I don't think that is really well understood quite yet. Especially like when we're talking about, okay, we are now working with contemporary samples that we are making ourselves. We're studying the interaction of plant chemistry versus color. But the direction where we do want to go in the end is to study the historical textiles. We do want to have a good understanding of how the tannins behave in our contemporary experiments before we start analyzing the historical samples. So if we do encounter, similar chemical profiles in the historical sample, we might be able to trace that back in a way to the plants that were used in the dyeing. And that doesn't go just for the tannins, but also for the other polyphenols as well. Hopefully that answers the question at least.
So now we want to get into two final questions for you.
Yeah.
And it's a question that I've asked many of your colleagues on this project and our colleagues. My first one is, what do you think is the future of natural dyes and the coloration of textiles?
That is a good question, and we are now very much leaving like my comfort zone and area of expertise here. And this is definitely, like, I have no trouble admitting that this is very much a topic that I had never thought about before joining the project, like chemistry of natural dyes in dyeing textiles. Like, the more part of this project, the more I'm learning that there's actually issues that these compounds and these natural sources could help solving. Especially after I've seen the stuff that people can make with the natural dye sources, and the color palettes that they can create. Like, there's definitely something there, I feel. And at least, if I consider myself, knowing, that my t-shirt or whatever, if it's dyed using plants or something like that, it brings me so close to, my passion in a way.
Yes.
So, I would be very happy to wear something like that. It would bring me, I don't know if a joy is a good word, but it would bring me some kind of connection to the, plants and the plant chemistry.
Yeah.
Maybe there's something there as well. Maybe it would help people to root to nature a bit more. If they would have clothing or other textiles that are plant dyed, something that can be done more locally.
I think that ties into, because one of the things that I've seen over the course of the project that you just touched on is the idea that by the end of this, we're not only going to know more about tannins, we're going to know what parts of nature have that it can be utilized, but we better understand nature. We then understand what we can utilize from nature. And I think for the future, for sustainability, relying on nature, but also making sure that when we do extract those, that it doesn't affect the conservation of nature, that nature is always going to be there.
That's something being natural doesn't make it better.
And it doesn't mean it's always going to be there.
No.
And I think having this information allows us to understand maybe even develop, not synthetic, but develop, I know it's not yours, but to develop molecules or compounds that allows this to happen where we're not utilizing nature and we're protecting a little bit more.
Yeah, just like pure ideologically, the idea of having plant-dyed clothes and textiles, for me it's very appealing.
Yes.
Of course, the road is long, to achieve that, but, just like...
It's not impossible, but the road is long.
No, it's
Absolutely.
not impossible, but it, just from like ideological point of view, it's an idea that I find nice.
Yeah. And you're contributing to that. And that's the fantastic part of it.
In some way, maybe.
Yes.
Yeah, in some way, hopefully.
We're all cogs in a big machine.
Yeah.
So our final question, and it's not really related to research, it's just your kind of opinion. What does color mean to you?
Yeah, this is another rough one. Like for color is something that I enjoy, especially in the nature. Like I'm the type of guy, like if you go through my wardrobe, you find black t-shirts, you find white t-shirts, you find black and white striped t-shirts. So like color is not something that I express very much daily, but it is definitely something that I like enjoy.
Okay. That's an interesting take because most of the people that when we've asked this question, it's about identity, expression, communicating.
Yeah, for me, it's not really that close thing to me. Like I do think about color in that way as well, but it's mainly like if I think about color and what in color gives me like joy, for instance, it's the colors that I see out there, like autumn time, for instance. It's amazing time because of all the colors that you can see everywhere. Every spring, I always enjoy it when you start seeing loads of green everywhere and you have these lush trees, growing and all that. I very much enjoy that. And also during wintertime, maybe not here in Helsinki, but in Rovaniemi, in the wintertime when everything is white.
Yeah.
That's also amazing.
I've loved that more in Finland than I have in other places.
Oh, really?
I think there's something about Finland, in my opinion, that the snow is more pristine in many ways because I've been in rural Pori.
Yeah.
I've been to Rovaniemi, and oftentimes the snow is not that disturbed. So it's pure, it's clean, and when you're looking out over a field of just pure, clean white snow, it's beautiful, it's bright, it's like But what's interesting is it's devoid of color. And which is interesting to me because it's beautiful, but we go from fall, and it's funny because you said, oh, I like when spring, the green that comes out in the spring, but I also love the fall. Then it's like a whole period of white.
Yeah.
And maybe that's why we appreciate the colors. Yeah. But I get
maybe
what you're saying.
Maybe that resets your palette, for the spring then.
I love that, resetting the palette,
Yeah, especially in Lapland where, you have a lot of snow. Winters, like here in the south, in the coastal region, they can be tough when you don't have it. Yeah. So you only have the darkness. That's not something that I enjoy. But especially in the north where you have a lot of snow, even like the white is just amazing. And it's after the white, comes then the spring and the green. And all the colors that come with it. Yeah.
It's completely different from where I come from, where one snowflake dropping in Georgia will halt traffic every single time. We don't see that much white. We don't see that. But it's an interesting cultural difference that we see across the world and the way that some people in different parts of the world see color than others. Yeah. And I think in Finland, there's much more of an appreciation for white and for black because that's used a lot. And there is a appreciation for color. And I think for you, it's more of the visibility of seeing it in nature, but also I wouldn't say it's an aesthetic thing because it's not what you wear, but it's something about reassuring about seeing it out in nature that connects you to that.
Like color has been a big part of my professional past as well, in a way, because before this project I did work with the red wines. I made a PhD thesis about red wines, and you really cannot escape the color when it comes to red wine. In a way, like color has been also a professional interest for quite some time. Yeah, it's also a big puzzle for me. It was with the red wines, and now it's with this plant dyeing.
Well, I want to thank you. This has been fantastic, and we will leave all the information in for our followers to listening to this about the exhibition and also information about you as well. Thank you, Juuso.
Thank you, Justin, for having me.